Hey there, welcome to the first part of Chapter 23, Evolutionary Processes. By the end of this chapter, you guys should be able to apply the Hardy-Weinberg principle to determine if natural selection is occurring on a particular gene. You should be able to tell me about the four evolutionary processes, including how these processes affect genetic variation in the population and average fitness in a population. You should also be able to explain how non-random mating can affect genotype frequencies without actually contributing to evolution. Oh, by the way, I'm going to talk about these first two objectives in part one, and then the whole rest of the chapter will be in part two. Next, you should be able to contrast directional selection, stabilizing selection, disruptive selection, and balancing selection. Don't worry, you'll know what that is when we get to it. You should be able to explain sexual selection and discuss why it tends to affect one sex more than others. You should be able to explain genetic drift and describe the circumstances under which it is likely to cause evolution in natural populations. You should be able to explain gene flow and its effect on populations. And you should be able to explain the importance of mutation to evolution. All right, so just a reminder, when we're talking about evolution, we are talking about changes in allele frequencies. All right, let's make sure everyone remembers the difference between an allele and a genotype. All right, if I say big A, what's that?
That's an allele.
Okay, good. What if I say big A, big A?
Oh, that's a genotype.
Yeah, okay, you guys got it. So we're looking at changes in allele frequencies, the proportion of the population that's big A versus little a. Okay, so that is what we mean by evolution. That is driven by four processes that we're going to go through one at a time. The first process is natural selection. So natural selection is going to increase the frequency of alleles that contribute to reproductive success in a particular environment. In other words, natural selection causes alleles that help individuals survive and reproduce. Those alleles will become more frequent in the population over time, right? Second is genetic drift, and I'll talk more about what this is in a minute, but this is a process that causes allele frequencies to change randomly, not because they're better or worse, just randomly. Third, gene flow occurs when individuals leave one population and join another and then breed, okay, like migration, etc. And then the fourth one is mutation, and mutation modifies allele frequencies by continually introducing new alleles. All right? So just to make sure everyone's on the same page here, evolution by natural selection is a phrase we say a lot. What we mean is change in allele frequencies caused by natural selection. We could have evolution by genetic drift, right? We could have evolution by gene flow, and we can have evolution by mutation. All right, get it? So these are four processes here that contribute to evolution. All right, so if you think evolution is happening in a population, how can you tell? And so basically what you're going to try and look at is whether or not the allele frequencies in a population are changing. Are there more big A's than there were in the past? What you need is a null hypothesis. This is a hypothesis that describes what should be seen if the hypothesis that you're testing is not correct. So in this case, the null hypothesis is a population with no evolution. The null hypothesis is a population where allele frequencies are not changing. And the null hypothesis for no evolution is the Hardy-Weinberg principle. All right, so in order to talk about the Hardy-Weinberg principle, we need to introduce the idea of a gene pool. So a gene pool is a kind of a tool that lets us measure allele frequencies in a population. So if all you're interested in is how many big A's there are over time and how many little A's there are over time, then who cares whether those big A's come from a big A individual, a big A little a individual, okay? So when we want to think about how all of these alleles contribute to the next generation, we will assume that the pairs of alleles that go into the gametes, meaning the sperm and the egg, are picked randomly from the pool. So if you remember, if this individual is big A, what alleles are possible in its sperm?
Big A.
Yeah, what else?
It could be a big A also, because that's all there is.
All right, good. What about this one?
Well, they could give a big A or they could give a little A.
Yeah, okay. All right, so all of these alleles that are locked into the genotypes of the different individuals can all just get dumped into the pool that's going to make up all of the sperm and eggs that are contributing to make the next generation. Okay? So we can basically take all of these alleles that are locked up in the genotypes and just kind of dump them in here. Okay? All right, so in this particular squirrely example, what proportion of alleles in the gene pool are big A?
I can't even look at it. The squirrels are so distracting.
I know, dog. Just look away for a moment. Anyone else? Bird, you don't care about squirrels. Why don't you tell me what proportion of alleles in the gene pool are big A?
Okay. 1, 2, 3, 4, 5, 6, 7, 8. Wait. 9 out of 12.
Yeah, 9 out of 12, right? 1, 2, 3, 4, 5, 6, 7, 8, 9 out of 12. All right, so
That's 3/4.
yeah, that's 3/4. So 3/4 of the alleles in the gene pool are big A, right? And we basically can see the same thing if we look at the whole animals, right? 1, 2, 3, 4, 5, 6, 7, 8, 9 out of 12 of the alleles are big A. All right. What proportion are little a?
Oh, that's like 3 out of 12.
Yeah, so 1/4. All right, so what is the probability that we pick two A's to make big A offspring? All right, so let's put a pin in that for a minute and talk about some rules of probability, okay? Yay, rules of probability. All right, the first rule of probability is called the AND rule. And in some books, they call this the product rule, right? Product meaning multiply. And so this rule of probability says, if you want to know what is the probability of both A and B happening, you need to multiply the probability of A happening and the probability of B happening, okay? So that's the AND rule. So if you want to say what's the probability of this and that happening, you multiply the two probabilities. All right, the other rule is the OR rule, sometimes known as the sum rule. And so if your question is asking what's the probability of either A or B happening, then you're going to add the two probabilities together. So like, for example, what's the probability of either flipping a heads on a coin, or flipping a tails on a coin? So what's the probability of flipping a heads?
Half.
Yeah. What's the probability of flipping a tails?
That's also a half.
Yeah. So what's the probability of either flipping a heads or the tails?
100%, because that's all there is.
Yeah, so you would add them, 0.5 plus 0.5, right? Okay, so those are your rules of probability. All right, so now let's kind of work this out with the squirrels, how we would do this. Well, maybe we can go back a slide too. So now that you know the rules of probability, what's the probability that we pick two A's to make big A offspring? So what's the probability that we pick one big A?
Oh, it's 3/4.
Yeah, it's 3/4, right? What's the probability that we pick an A again? What's the probability we pick an A? Just a second time.
Oh, it's also 3/4.
All right, so what's the probability that we pick one big A and another big A?
It's 3/4 times 3/4.
Yeah. Okay, so let's try and do a little bit of a problem about this. So remember I introduced you to the 6 kinds of population genetics problems, right? So if I just told you the number of squirrels who had each genotype, we went out and we genotyped them all, and we found out that 320 of the squirrels out of 500 are big A, 160 were big A, little a, and 20 were little a. What are the genotype frequencies? So how do you figure this out? Oh, I know. It's just like the number of each genotype out of the total number of individuals. Yeah, right? This is just a simple math problem. Like if somebody asked you what's the proportion of individuals with black shoes on, you would see how many people had black shoes and then how many people there were total, right? It's exactly the same kind of problem. All right, so what math are we doing here?
It's 320 divided by 500.
Yeah, and that divides to be 0.64. All right, what is the fraction that we need to figure out for big A, little a?
160 divided by 500.
Yeah. And little a? Someone else?
Yeah, that's just 20 out of 500.
Yeah. Okay. So these are our genotype frequencies. Again, when we're saying genotype frequency, we want to know how frequent big A, big A, big A, and little a are in the population. So big A is found with a frequency of 0.64, big A, little a is found with a frequency of 0.32, and little a is found with a frequency of 0.04. Okay? There are only these three possible genotypes at this particular gene locus, right? So this and this and this have to add up to 1, okay? Because there's no other choices. So I would always recommend, after you do a calculation like this, just add these up and make sure they equal 1. All right, so now we need to figure out the number of alleles in the gene pool, okay? So there's a couple of different ways to calculate this. Can you think of different ways to calculate this? Yeah, you could like just add up how many like big A's there are in the big A ones and then add it to the number of big A's in the big A little a ones over the total number of alleles. Yeah, you could totally do that. And then there's a second way you could do it that's just pretty close to the first one.
Oh, I know. What I like to do is I would do like the number of individuals who are big A, they only give big A, so I would use that number plus half of the number of the big A, little a individuals.
Yeah, so that's also possible. So let's see, what do we do here? Well, actually, yep, so the number of alleles that they have in the gene pool, these guys are contributing 640 big A's. These guys are contributing 160 big A's and 160 little a's. And these ones are contributing 40 little a's. All right? How many alleles are there total in the gene pool anyway?
Oh, I know, it's 1,000.
Yeah, it's 1,000, right? Because there's 500 squirrels and they each have two. One that they inherited from their mom and one from their dad. All right, so let's figure out the frequency of big A using one of those two methods. Okay? All right, do you guys want to each work out using your own method? So you up there in the corner of the ceiling here, you want to do the one where you do 2 times 640 plus 160 over 1,000? Okay. All right. Bird, do you want to do the other one where you do 640 plus half of 160?
Okay, I'll do that one.
All right. So I just have it written out. The second way, the first way, where you have 640 plus 160 divided by 1,000. Just a word, make sure that your denominator is correct. If you do this kind of calculation, make sure you're not dividing by 500, right? Because there's not 500 alleles total in the population, there's 1,000. Alternatively, if you had done 640 plus a half times 160 divided by 500, you would have gotten the same answer. All right. So how do you calculate out the allele frequency of little a? I do like 20 times 2, which is 40 plus 160 divided by 500 times 2 is 1,000. Yeah? And you get 0.2? Yeah. All right, and just note that all the alleles in the population, if they're not big A, they got to be little b. If they're not big A, they got to be little a, and if they're not little a, they got to be big A, right? So these two numbers have to add up to what?
They add up to 1.
Yeah, they add up to 1. Okay. So let's consider the gene pool of this population of 500 squirrels. So we figured out what the genotype frequencies are, and then we just calculated out the allele frequencies.
We did it!
We did it. Okay. And then I just pointed out to you that the genotype frequencies have to add up to 1, because if you're not big A, and big A, you got to be little a, right? And so if these are the only 3 options. In terms of alleles, these are the only 2 options, so again, these have to add up to 1 as well. All right, we have a name for the frequency of the dominant allele, and we call that one P, okay? And so we say that this value here equals P, right? The frequency of big A alleles equals 0.8, which equals P. The frequency of the recessive allele is named Q, so the frequency of little a is 0.2, is Q. All right, so let's just say that we wanted to make a prediction about the future. We wanted to know what the frequencies were of each genotype in P's and Q's, okay? So if you're big A, you are the result of a big A egg and a big A sperm coming together, right? So it's the probability of inheriting a big A and the probability of inheriting another big A. So what do you do if you hear and?
You multiply.
Yeah, you multiply, okay? And so what's another name for P times P?
Oh, that's P squared.
Yeah, P squared, okay? All right, so, whoops, I forget which square comes up next. So what's the probability of inheriting a Q from one parent and a Q from another parent?
That would be Q times Q.
Yeah, Q times Q, or Q squared. What about inheriting a big A egg from your mom and a little a sperm from your dad?
Oh, that's P times Q.
Yeah, what about inheriting a little a egg from your mom and a big A sperm from your dad? Oh, that's P times Q again. Okay. And so then we have other names that we just said. P times P is P squared, right? P times Q is PQ. Q times Q is Q squared. Okay? And we don't really care whether you got the egg from your mom and the sperm from your dad, big A from your mom, or whether you got the big A from your dad. It doesn't make any difference. So we can maybe add these two categories together because they both result in offspring that are big A, little a. So this gives us the Hardy-Weinberg equation. And the fact that it equals 1 isn't that interesting. So people will often look at this and say, oh, it equaled 1, so the population is in equilibrium. That is not how you figure out if a population is in equilibrium. The only thing that having it equal 1 means is that you did the math correctly, okay? So you have to be, there's only 3 genotypes in the population, big A, big A, big A, little a, and little a, little a, so they always have to add up to 1, okay? So that's not, but it's just kind of the way we tend to write this equation. So please do not tell me that a population is in equilibrium because P squared plus 2PQ plus Q squared equals 1, okay? Okay. One thing that's important to notice is that each generation, alleles are going to be transmitted from parents to offspring, right? So, meiosis. What's meiosis? What's meiosis? Anyone?
Oh, yeah, that's when you, make special cells that have half the number of chromosomes of your other cells, and so they get, one of your 2 copies of alleles.
Yeah. So that's meiosis, right? We use meiosis to make sperm and eggs if we're diploid. All right. So meiosis and random combinations of gametes, meaning which sperm you get and which egg you get, are going to shuffle around all of the alleles in the population to make the different genotypes, right? But it should be noted that if no evolution is happening, allele frequency should not change. So if you started out with the frequency of big A being 0.8 and the frequency of little a being 0.2, after everyone mates with each other, if there's no evolution happening, you should end up with the frequency of big A being 0.8 again and the frequency of little a being 0.2 again, right? So it's like shuffling the cards in a deck, okay? Meiosis and the random combination of gametes shuffle all the cards in a deck, but they don't make a change in the allele frequencies in the population, okay? All right, so let's do a little bit of a problem here. So remember that little population of squirrels from before? The question you want to know is, are they in Hardy-Weinberg equilibrium? And what you're really trying to figure out is, are they a population in which no evolution is happening? All right, so the first thing we need to do is calculate out the allele frequencies of big A and little a using just regular math, okay? Then we're going to use the allele frequencies and the Hardy-Weinberg equation to determine the genotype frequencies that you would predict if the population is in Hardy-Weinberg equilibrium. So let's do this first part over here, okay? So let's figure out the frequency of big A. All right, so what would we do?
Oh, I would take the 0.667 and then add it to half of the 0.167 for the big A little a, because the big A ones, 100% of them are big A's, but the big A little ones, they're only half big A's.
Okay, so that should give you a big A. All right, do you want to come write it up on the board here? Oh, dog.
I tried to write it on the board.
All right, that didn't work. All right, bird, did you do it?
Yeah, I wrote it up on the board.
Okay, so for big A, the frequency of big A, 100% of the alleles in big A individuals are big A's, right? So we just did 0.667, or you can imagine that's multiplied by a 1, for 100%. And the big A, little a individuals also have big A's, but their genotype frequency is 0.167, and like half of that is made out of big A's and half is made out of little a's, right? So if you multiply 0.167 by half and then add it, right? Because we're looking for big A's that are in either big A individuals or big A, little a individuals, right? So we're adding those, and we get 0.75. For little a, let's see. So then we find little a's in big A, little a individuals and in little a individuals. Okay, and we're willing to use big little a's that are in either big A, little a individuals or big A or little a individuals. So these little a individuals are 100% little a. Okay, these big A, little a individuals are half little a. So we multiply that by half, and that gives us 0.25. Okay. So these add up to 1, so we did our math right.
Yeah, I know. I checked.
Good job. All right, so now we know the allele frequencies in the parents, right? So let's figure out, using the Hardy-Weinberg equation, to figure out if the genotype frequencies in the parents match the genotype frequencies in their offspring, okay? Or even in the parents themselves if the population is in Hardy-Weinberg equilibrium, either thing. All right, so how do we calculate out the frequency of big A? Well, let's think for a second. For the frequency of big A, what's another name for that?
That's P.
Yeah. All right, the frequency of little a, what's that?
That's Q.
Yeah, that's Q. Okay. So we can use that P and Q to figure out what we would predict the genotype frequencies should be if the population is in Hardy-Weinberg equilibrium. So what is the frequency of big A if the population is in Hardy-Weinberg equilibrium?
Oh, I know. It's P squared.
Yeah, it's P squared, right? What is the frequency of big A little a if the population is in Hardy-Weinberg equilibrium?
Oh, that's 2PQ.
Yeah, that's 2PQ. And what's the frequency of little a if the population is in Hardy-Weinberg equilibrium?
That's Q squared.
Yeah, that's Q squared. Okay, so let's do that. So the frequency of big A is P squared, right? So it's P times P, and that is 0.565. The frequency of big A little a is 2 times P times Q, and the frequency of little a is, let's see, where are we at? Is Q times Q squared. Okay. All right, so this gives us the frequency of big A little a, and little a that we would predict using Hardy-Weinberg. All right, whoops, forgot those. Okay, so this is the frequency, these are the frequencies that we just calculated, right? In the previous problem, see, there we go. All right, so there they are. So let's compare the genotype frequencies that we predicted using Hardy-Weinberg to the genotype frequencies that we predicted, or that we found actually just measuring it in the population. So what we actually found by measuring was that the frequency of big A in the squirrels was 0.667. The frequency that we would have predicted based on Hardy-Weinberg is 0.563. Okay? The frequency of big A little a that we measured on squirrels was 0.167, and what we actually found was 0.375. And what we actually measured was 1.167, and what we found was 0.063. So are they the same?
No, they're not very close.
Yeah, they're not very close, right? You find that we have the same frequency of big A and little a in the actual population, but we would have predicted we would have a lot more big A and fewer little a, right?
Yeah.
All right, so again, we talked about the Hardy-Weinberg principle as being a way to predict what happens if there's no evolution, right? So if there's no evolution, we have to expect certain conditions, right? So these conditions are the 5 assumptions of the Hardy-Weinberg principle. The first is that there's random mating in our population, right? There's no mate choice, and gametes will combine randomly. If we don't have random mating, actually this will not affect allele frequencies, but it will affect genotype frequencies. For more information on that, take my class on evolution. All right, for number 2, there's no natural selection, right? All individuals contribute equally to the gene pool. There are not some individuals who survive better and have more babies than other individuals and then leave more genes. Third, there's no genetic drift. So this means that the alleles are picked by chance. And we're going to talk more about this later, but when you have very, very small populations, you're more likely to accidentally have your alleles not represented. So in order to avoid genetic drift, you have to have a very, very large population. So sometimes you see this assumption written as no genetic drift, and sometimes you see this assumption as the population must be large. All right, the fourth one is no gene flow, so you can't have new alleles added or lost from the gene pool. So if you had a nearby population and big A individuals were coming in all the time, over time you would end up with more big A's, right? So we can't have that. And then fifth, there's no mutation. And so this is the idea that you're not getting new alleles introduced into the gene pool because of mutation. So for example, if little a's were very likely to mutate into big A's for some molecular biology reason, you might end up with more big A's over time. All right, so if you violate any of these assumptions, evolution will occur. It's inevitable, okay? All right, so let's do a couple of examples. So in humans, we have, the ABO blood groups, which you've heard of, but we also have MN blood groups. And so MNs are different kinds of antigens that are on the outside of our red blood cells. So we have little antigens on the outside of the red blood cells. And so let's see here. So there are the alleles M and the allele N, and these both code for red blood cell surface proteins, little antigens that stick on the outside of your red blood cells. And M and N are codominant, right? So do you remember what codominance is from 1113?
Oh, I remember that one. That's when both alleles are expressed at the same time.
Yeah, both alleles are expressed at the same time. Okay. So M and N are codominant, and what that means functionally is if your genotype is MM, then you just have lots of M antigens or proteins on the outside of your red blood cells. If your genotype is NN, you'll just have the N antigens on the outside, and if you're MN, you express the genes to make both the M and the N antigens, so you have both, okay? All right. So those are your genes. So your genotype could be MM, MN, or NN, and then your phenotype is that you have either M antigens, N antigens, or both. So people were curious to know whether MN blood types, or the alleles for MN blood types, are in Hardy-Weinberg equilibrium. Okay, so this problem I'm going to give you is kind of like problem 5. Remember in lab I gave you 6 types of problems? So this is like problem 5 of the 6 types of problems. This is a problem in which I give you the genotype frequency of the parents, and you tell me the predicted genotype frequencies of the offspring, assuming that the population is in Hardy-Weinberg equilibrium. Hint, make sure that you first use regular math to calculate out the allele frequencies in the parents, and then you use Hardy-Weinberg to figure out the offspring genotype frequencies. So it's a 2-step process. All right, so this comes from a very old paper when people first understood the blood groups and they first had some idea about evolution in traits. Quantitative genetic kind of methods like we're using to look at differences among traits. They went and looked at blood types. So they found that in kind of white people in the U.S., they observed a frequency of MM at 0.92, MN 0.494, and NN 0.213. Okay, And from that, they calculated out the allele frequencies of 0.54 of M and 0.46 of N. And then they used Hardy-Weinberg. So they figured out, okay, they would expect that P squared people would be MN, so 0.54 squared, right? They predicted that Q squared people would be NN, so 0.46 squared, and then they predicted 2PQ people would be MN, so 2 times this, okay? All right, so we can compare. So in Caucasians in the US, the predicted and the observed values are very similar, right? Okay, so it doesn't look like evolution's happening. In Australian Aboriginal people, they had observed these genotype frequencies. They calculated out the alleles, and then they calculated out the expected genotype frequencies if the populations were in Hardy-Weinberg equilibrium. So does that match?
I mean, sort of. It's a little bit off, but it kind of matches.
Yeah, so we'd have to use statistics to determine if it's different enough to be considered different. Okay, so now we're going to work on a minority group in Japan called the Ainu. And what they went and observed from blood typing people is that the frequency of MM is 0.179, frequency of MN is 0.502, and the frequency of NN is 0.319. Okay, that's what they measured. All right, the next step is to figure out the allele frequencies. All right, so I'm going to let you guys calculate this out on your own. Okay, so somebody tell me what math you would do to figure out the frequency of M.
Oh, I would take like 0.179 and I'd add it to half of 0.502.
Okay, good. And so then what would you calculate out somebody else to get N?
Okay, I don't know. Okay, I would take 0.319 and then I add it to half of 0.502.
Okay, so if you did that, you'd get an allele frequency, a frequency of M of 0.430 and a frequency of N of 0.570. Okay?
Great.
Great. All right. And we didn't use any kind of Hardy-Weinberg to figure those out, right? That's just a straight up regular math kind of a problem. All right, now we need to calculate out what we would expect the population genotype frequencies to be under Hardy-Weinberg.
So now we use P's and Q's?
So now you use P's and Q's.
Great.
All right, so which of these things is P and which is Q?
The 0.430 is P and the 0.57 is Q.
Yeah. All right, so in P's and Q's, what calculation would we do to figure out the expected genotype frequency of MM?
That's P squared.
Yeah. And what about, so P squared would be?
0.430 squared.
Yeah. All right. What about MN? What calculation would we do?
That's 2PQ.
Yeah, it's 2PQ. So what would you multiply?
2 times 0.430 times 0.57.
Okay. And then NN, what is that in P's and Q's?
That's Q squared.
Yeah, so what's that?
0.57 squared.
Okay, good. That's what we get. All right, let's compare. So, are the expected genotype frequencies the same or different than the observed ones?
They're looking kind of different, but not quite sure.
Yeah, it's a little bit hard to tell. We would probably need to use a chi-squared test to determine if the difference between the expected and the observed was different enough that we would consider that that population was, the expectations for Hardy-Weinberg equilibrium were not the same as the observed values, right? So let's just review one thing. So if the expected and observed are the same, has evolution happened?
No.
Right. If the expected and observed are different, has evolution happened?
Yes.
Okay, good. All right, so the observed MN genotype frequencies were almost identical to the MN genotype frequencies expected by Hardy-Weinberg. So we would say that and so just pretend I went and did the chi-squared for you and I didn't find a difference, right? So the genotypes in the MN locus do seem to be in Hardy-Weinberg equilibrium. So the M and the N alleles are not changing in frequency. So you would say that evolution is not happening. And so because evolution is not happening, then we can say that the MN blood groups are not being affected by any of the 4 evolutionary processes. You guys want to remind me what the 4 evolutionary processes are?
Yeah, natural selection.
Yeah, natural selection. What else?
Mutation.
Yeah, mutation. What else?
Genetic drift.
Yeah. And what's the other one?
Gene flow.
Yeah, gene flow. Okay. So we know that there's not natural selection, there's not mutation, there's not genetic drift, and there's not gene flow happening in terms of the gene locus that contributes to the MN blood group. Okay? All right, let's look at a different set of alleles. The question is whether HLA alleles are in Hardy-Weinberg equilibrium. HLA, well, the HLA family of alleles, including HLA-A and HLA-B, code for major histocompatibility, or MHC proteins. So these are proteins that all mammals have that help us recognize different disease-causing organisms, okay? So they're part of our immune system. So the hypothesis was that potentially HLA and HLA-B heterozygotes, and again, these represent, I should point out, these are not 2 alleles at one locus, these are 2 different gene loci, okay? So you got like a big A and a little a over here, and a big A and a big B and a little b over here, okay? So the hypothesis is that HLA and HLB heterozygotes might be more fit than homozygotes, and the reasoning behind that is if you're big A, little a, or big B and little b, you have more different kinds of major histocompatibility proteins, so you can produce a wider variety of, or you can identify a wider variety of bacteria and viruses than a homozygote who has fewer different kinds of HLA proteins. All right, so to test this hypothesis, researchers went to the Havasupai tribe in the Grand Canyon, and they determined the genotypes of 122 people. All right, and this is what they found. So for the first locus, the HLA-A locus, they observed, oh, and by the way, these results look a little bit different than what I just showed you. So because our hypothesis is about differences between homozygotes and heterozygotes, they added together big A, and little a, right? So, all right, so there's 38 big A, and little a individuals. There's 84 big A, little a individuals. If you used Hardy-Weinberg equilibrium principle, we would expect that 48 individuals would be homozygotes and 74 would be heterozygotes, okay? Looking at the HLA-B gene locus, they observed that 21 individuals were big A, and little a, and 101 were big A, little a. Compared to what they would have expected, they would have expected 30 of them to be homozygotes, big A, and little a, and 92 of them to be B's. They expected 30 of them to be big B, and little b, and 92 of them to be big B, little b. Okay? So what do you guys think? Do the expected genotype frequencies match the observed?
Yeah, I don't think so. It's pretty different.
Yeah, in which way is it different?
There's more observed heterozygotes than expected so there's more heterozygotes observed than expected, and then there's fewer homozygotes observed than expected.
Yeah. So they do not match. So if we do the stats on that, this confirms they do not match. Because they do not match, we can tell that at least one Hardy-Weinberg assumption has been violated here, okay? So we don't know which of the 5 it is, but we can come up with hypotheses about which of the 5 assumptions was violated and then test them. So here's a couple of the hypotheses. One of them is that perhaps mating is not random with respect to HLA genotype. It is possible that people prefer partners with different genotypes than themselves. And the reason why we think this might happen is because we know in experiments on mice, if you give a female mouse a male who is either the same HLA type as she is or a different HLA type, she will always choose the male who's different. It's not like she can go smell him and say, oh, I know you're a different HLA type. Instead, she just looks at him and thinks, I like this guy. I like him better than that guy. Okay? Same with people. We're not saying that people can go and figure out whether other individuals are the same HLA type or a different HLA type from themselves. Just that because this is basically a homology that we may have inherited from our rodent ancestor, we just like particular people more than other people. And one reason why we might like somebody romantically, be attracted to them, might be because they differ from us in their HLA alleles. So that's one hypothesis. A second hypothesis is that potentially these heterozygous individuals have higher fitness. And so for that reason, there are just more living heterozygotes in the population. And the reason why we think this might be true is that in a different population, married women who had the same HLA alleles as their husbands had more trouble maintaining pregnancies. So it is possible that babies that are heterozygotes are more likely to survive to adulthood than babies that are homozygotes. But these two hypotheses would have to be tested in this particular population if we want to be able to demonstrate why this population is not in Hardy-Weinberg equilibrium.